Okay, welcome everyone. We're going to resume this afternoon section. I'm the chairperson, Professor Kenneth Learn, the Director of State Key Laboratory of Marine Pollution. First of all, I would like to invite Dr. Daisy Pandayo. She is representing PEMSEA. She would like to say a few words. Daisy, please.
Thank you, Professor Leong, and good afternoon, everyone. My name is Daisy Padayaw, so I'm with PEMSEA, the Partnerships of Environmental Management for the Seas of East Asia. First of all, I would like, on behalf of PEMSEA, I would like to express our sincere gratitude to the State Key Laboratory of Marine Pollution, the City University of Hong Kong, the Department of Applied Biology and Chemical Technology, the Hong Kong Polytechnic University, the Department of Science and Environmental Studies, the Education University of Hong Kong, for organizing this interesting and very informative training session on marine environmental research. And of course, I would like also to express our appreciation to all the participants for joining us today, and hopefully you can also join us tomorrow. This session is part of the pre-events for the East Asian Seas Congress, which will culminate on December 1 and 2. So we hope everyone can also join us during the culminating activity on December 1 and 2. And this morning, we have listened to some of the researches of the different universities in Hong Kong, like one of them, like the one that was presented on microplastics. And James Fang said that the university or his institute, or maybe personally, maybe their institute are open for collaboration. So hopefully we can forge possible collaboration with the universities of Hong Kong to share their expertise and knowledge to our participants from the region. And yeah, thank you very much. And I look forward together with the participants for a more interesting discussions this afternoon. Thank you, Professor.
Thank you, Daisy. Thank you for the sharing. We would like to thank her support and the support from PEMSEA for co-organizing this collab event. So this morning we have a maximum of 65 participants joining us this morning. So we hope the number will be increased over time. Right now we have 43 people, very good. So may I introduce to you the first speaker, Professor Wutofu. He was the founding director of the Center of Excellence in Marine Environmental Research and Innovative Technology in Hong Kong. We call it MERIT. And then subsequently, he also established the State Key Laboratory of Marine Pollution based on the same team of MERIT. And then in 2008, he successfully applied the status for the MERIT Center as the Regional Center of Excellence in Marine Pollution in East Asia Seas region. So for those who missed the morning section, he already talked about the pollution monitoring consideration. Now he moved on to talk about marine conservation and environmental management. It's a kind of new paradigm shift, some new ideas he wants to share with you. So may I give the floor to Professor Wu, please?
Yeah, thank you, Kenneth. Can you see the slides?
Yes, okay, no problem, very good.
Okay, okay, good afternoon, everybody. Welcome back after lunch. And this afternoon, I'll talk about the conservation and environmental management, a paradigm shift. So I'm trying to tell you the new concepts in addition to what we used to know about conservation and marine conservation and environmental management, and hope to have some insight on the environmental management in the near future. Now, as you may probably aware, and then human population increase, overexploitation of natural resources, habitat degradation, pollution, and biological invasion, climate change, and so on and so forth, all lead to drastic environmental damages and environmental disaster, including species extinction and the decomposition of and ecosystem failure and so on and so forth over the world. So it become a major problem nowadays, and in particular, the effect caused by climate change has become a hot topic nowadays. Now, as such, the Convention of Biodiversity aims to conserve biological diversity and sustain the use of these biological components. In 1993, China became a signatory to the CBD, and then Hong Kong joined them in 2001. Now, that provokes our new way of thinking. Say, in the early days, what we learned from the Convention on Biological Diversity, and in most of the literature, and in most of the environmental protection things, and then they aim to protect all species in our ecosystem whenever and wherever possible. Well, that's pretty holy, and I guess nobody would dispute that if possible, we should protect all species in the ecosystem and make a better environment for ourselves. And there's no dispute at all. But the question is that, is this possible? Now, in some place, like the sort of, for example, in Great Barrier Reef, no problem at all, because there's not much anthropogenic activities, and then the environment keep us something like 50 years ago. But even so, and we are facing the problem of coral bleaching and things like that, and then for one reason or the other. Now, but in many places, if there's a pristine area, we keep it in the same way, and we have some difficulties. Not to mention that in many places in Hong Kong and many places in the Southeast Asia and elsewhere, and then we got lots of urbanized environment. So we have to learn how to live together with our environment harmoniously. Take Hong Kong as an example. Now, Hong Kong is a very small place. The total area of Hong Kong is only about 1,000 kilometers square, and yet it consists of all sorts of activity. Say, for example, we need to protect the Chinese white dolphin, which is the icon of Hong Kong, and then here, about here, and then we have to have a new airport. And then here, Victoria Harbour, we discharge the sewage water of something like 3.5 million people into this water. And then in this part, and then we urbanize, and we have yachting activities, swimming activities, and then fish culture activities, and so on and so forth. Now, the problem is that how do we deal with this situation? If we have conflicting use of the environment, of course, we then design different beneficial use in marine water of Hong Kong, saying that, okay, well, this part is for protection of the dolphin, this part is for human use, and this part is for swimming, this part is for monoculture, and so on and so forth. But is it okay? But when we have any development, it's quite often in Hong Kong, we got a major fight between the developer and green group. And in fact, it's not only in Hong Kong, it's fairly common anywhere in the world, that whenever you have development, you clash the conservation biologists with the green group, and then they say, go away, don't do it, and so on and so forth. So how do we live together? Now, so for environmental protection, what we do is that Now, the species from the species perspective. As I said before, can we protect all species? If yes, very good, no problem. We got no conflict and we live harmoniously with the environment. But if not, what is our priority? Now, so it's very common, of course, we have to protect the endangered species. Well, endangered species list on the red list of the International Union for the Conservation of Nature, like coral, like dolphin, like panda, black-faced spoonbills, and then in Hong Kong, we also protect the horseshoe crab and the Roman frog, and so on and so forth. These are endangered species. Then what else? Of course, we need to protect the species or the habitat of high conservation value, something like the mangrove and seagrass and coral reef and so on and so forth. Now, thirdly, we got to protect the species that are vulnerable to human activity. Some animals are very vulnerable to human activity, and then if you do something wrong, they'll extinct. Well, then we ask the question, is it enough? So we protect all this. These are pretty obvious, and then nobody would sort of dispute that we should not protect the coral, Chinese white dolphin, and panda. But is that enough? Now, then some of the guy, even include an academic, I wouldn't mention name, is that every single species is important. In this ecosystem, every single species is important, just like bolts and nuts of the alphabet. Losing any one of these bolts and nuts will lead to big disaster. So we must protect all species, and then sometimes all individuals. Then I scratch my head, is that true? Let me think about this. Now, of course, when we say you protect all species, in fact, in my opinion, it's very easy. Your 3 years small girl can do that, because just say no to any activities. So don't touch anything, don't do anything. No, so you protect all the species, right? You don't need a professional to tell you what to do. Now, but I can tell you that from a species perspective, we should not look in this way, and some species are more equals than the others. Of course, you may, most of you have read the book called Animal Farm by George Orwell, and then the story tells about something like the animals doesn't like human being. This is why they gang up together to have a revolution, and then they said all animals are equal. Okay, but after they grab power, and then they sort of they kick away the human being, and the animals become the boss, and they said all animals are equal, but they add on another appendix called, but some animals are more equal than others. In fact, that's true. In the ecosystem, some animals are actually more important than others. The first example is what we call the Keystone Species. Anybody been to a bridge in the UK or in Europe will know that there's a keystone here on the arch of the bridge, and there's a keystone. Now, you remove one of these break, does not matter. It's no good, but it's okay. But if you remove the keystone, the whole thing will fall apart. The same occurs in the ecosystem. A species that has a disproportionately large effect on its environment relative to its abundance. So we are not talking about the animals which are abundant in the ecosystem is important. They may occur in small numbers, but yet they are very important. Now, I cite you two examples. The first is the sea otters. They only have not a lot of sea otters along the western coast of America. The sea otters feed on sea urchin and also feed on the sea star. The sea urchin and the sea star feeds on this kelp, the macroalgae. Because of overfishing, the fishermen don't like the sea otters because they're competitive. They eat the fish as well, so they kill them off. But as the population drops, no, you can tell that immediately if there's less sea otter, there's less predator, the sea urchin and the sea star will begin to grow. And these two echinoderms feed on the macroalgae. So if they are abundant, basically they finish all the macroalgae. So the system is like this: with the sea otter, everything in good balance. Sea otter feeds on sea urchin and the starfish, and they in turn feed on the algae. Okay, so with the sea otters, they control the predators of the giant kelp and also the macroalgae. So the system is good. They support a lot of animals. But if you take off the sea otter, the sea otters, the sea urchin and the sea star begin to grow in great abundance, and they sort of finish all the sea kelp, and then a system like this. So eventually the whole system collapses, and then they support no other animals. Another very typical example is when you go to a rocky shore, you find this small animal called the limpets. Looks like an abalone, but it's not abalone. They do not occur in great abundance. You can find maybe 10 or 20 of them on a rocky shore. So you don't think that's important. But earlier on, way back in 1980s, and Professor Hawkins in the UK did a very simple experiment. He removed the limpets from the shore, and basically, after removing all the limpets, all the algae become the overgrowth. Because the overgrowth, they cover everything, so they kill off all the barnacles, mussels, and limpets, and so on and so forth. So you can see that if you disturb this, what we call the keystone species, can lead to dramatic changes in the entire ecosystem. Again, I'd like to emphasize these animals occur as one of the many members in the ecosystem. They do not occur in great abundance, but yet they are so important. If we remove them, the whole ecosystem will change, and then threatening the survival of the other species. Now, the other group is that what we call the functionally important species. Now, when we go to an ecosystem, we see all sorts of animals, but we actually know very little about what kind of function that they are providing and what kind of function that they perform in the ecosystem. Now, some species may play an important role in the function of the ecosystem, including controlling the productivity, energy flow, carbon fixation, or they may regulate the nutrient and biogeochemical cycles, oxygen and carbon dioxide balance, and then sometimes they form important link between different trophic levels. Now, again, to give you some example, that's a sort of general description of ecosystem. You got the consumer, producer, you got the composer. Most of people in the conservation, they focus on the consumers and the producers, the things that they can see. But in fact, a lot of these decomposers, you may not like to hear the cockroach is also important because they play an important role in nibbling these and the detritus and recycle them. Okay, and mushroom, and then this earthworm, they look ugly. These detritivores and decomposers, they look ugly and they're inconspicuous in the environment, and yet they are important, performing important function in this ecosystem. Now, another example is barnacles. You all see barnacles, plenty of them now on the rocky shore. And then the barnacles, you may like to know the barnacle is the #2 highest energy flow in the marine ecosystem. That is to say, they channel a lot of energy from the pelagic environment into the intertidal. And also when they spawn, they would produce a lot of larvae from the norpi and then go into the water. That is to say, the barnacles are important, not only energy flow in productivity of the rocky shore, At the same time, they are also important in coupling the benthic environment and the pelagic environment. So if you kill off the barnacles, although they look ugly, and then they grow rapidly, then however, you're disturbing the whole energy flow, making the whole productivity of the rocky shore decrease, and also you decouple the benthic and the pelagic and link of the environment. Now, so disturb these species, which are functionally important, will impair the ecosystem structure and function, and then leading to changes in the entire ecosystem, and then pose a significant threat to the whole ecosystem, to the ecosystem integrity, and then nobody can be escaped from them, and then all species would be affected. Now, so I hope you then agree with me that we should hire sort of we should accord the highest level of protection to these keystone species and the functional important species in the ecosystem, because any change in these keystone species or functional important species and all other species will be affected, and some may go extinct together. Now, if you look back to the very fundamental question, see why we need to protect our ecosystem from the human point of view is that we got to maintain different type of ecosystem services, which is desirable to mankind. That is to say, we're exploiting the environment in several ways. The first is that we need provisioning. We need the environment to provide food and natural resources to us. We need the environment to clean water and air for us. And then we need the environment to support a lot of this function that we want, like the crop pollination, energy flow, nutrient cycle, which are essential in our daily life. And then sometimes we need them to support the cultural services to us, like the recreational, we go diving, swimming, and then we go hiking and we see the mountains and things like that. Although they are not directly linked to us, but they provide good aesthetic value. And then, of course, we know that the environment provides a regulating function, control the climate and disease in many ways. This is why we need to protect our environment. We protect these ecosystem services. Now, coming back to this question, which I do not sort of agree, saying that every single species is important, just like bolts and nuts of an elephant. Losing anyone will lead to disaster. Now, if you look at this scientific side, the assumption from the ecosystem protection is that different species would perform different functions in the ecosystem, and then different species would interact with each other. And this is why we got to maintain a high diversity. There's no dispute on this. If you got a high diversity ecosystem, you got a high level of insurance to environmental disturbance. I'll come back to the mechanisms later. If you've got a high level of insurance to the environmental disturbance, you've got a high level of stability. That is to say that if you've got an ecosystem with high diversity, you'll be less vulnerable to human disturbance or to changes. And if you've got a low diversity ecosystem, this will affect ecosystem integrity and then an array of ecosystem function. Any change is more likely to decrease in the ecosystem stability and impair the desirable ecosystem services. That's the general principle. But let me show you an example. Now, that's you. You got 5 t-shirts, 3 pairs of shoes, and 1 trousers. If your good friend asks you, hey, John, can I borrow a t-shirt from you? Oh, sure, 1 t-shirt. And then the other guy comes in saying, can I borrow another one from you? You say, oh, okay. So you still got 3 t-shirts. And then another friend comes in saying, can I borrow a pair of shoes from you? Sure, you bear 1. You gave him 1. But if a guy came and said, can I borrow a pair of trousers from you? Oh, there's no way, right? Because the trousers is indispensable to you, whereas the t-shirt is dispensable because you got more t-shirts and then you can use. The same occur in the ecosystem. If you look at ecosystem function, now, this is a hypothetical example. You got two predators, A and B, feeding on the herbivore C, D, and E. So A feed on C and D, B feed on 3 of them, and then C feed on F and G, D feed on F, G, H, E feed on G, H, I, and so on and so forth. Now, if we remove A, what will happen? So basically, if we remove A, so you got less predatory pressure on C and D, right? And then B would be actually very happy because B is competing with you, right? And then without A, B can grow better, and then C can feed on C and D. So if you look at removing A, although it's not desirable, but however, the only change is that, well, A is eliminated, and then the whole ecosystem except A remain roughly the same. And then, in fact, sometimes B can also perform the function of A in terms of predation and controlling C, D, E, and F, G, H, I, and so on. Now, but if the same system, but different sort of linkage and different relationship, A feed on C and D, and then B feed on D and E, and then the plant herbivore F, G, H, I, and as the relationship as indicated by the arrow. So if we remove H, H is, well, one of the many plants in the system. If we remove H, what will happen is that immediately D got nothing to feed on because D feed on G, H exclusively, and same as E. So removing H means that you are also eliminating D and E. So we eliminating D and E, B will be also eliminated because B feed on D and E. Okay, A will be less affected because he would be feeding on C and F and G. And at the same time, if we remove H, because they are not competing for nutrient, and F or G may increase the abundance or decrease in abundance, and so on and so forth. Now, you can see as opposed to the previous example, removing A, removing H will cause major devastating effect in the ecosystem, change the ecosystem completely. Now, then in the last decade, a lot of people did study on looking at this, what they call the functional redundancy. Now, because according to the general belief, every single species in the ecosystem is important, and then because they contribute to the stability. But in fact, if high level of redundancy occur within the same functional group, the studies find that further increase in diversity may not necessarily contribute to high ecosystem stability. And then for several study in the terrestrial system, they show that nutrient retention, okay, may be saturated at relatively low level of diversity. That is to say, provided you've got certain level of diversity, further increase in the species richness in diversity does not add on the ecosystem function at all, and does not add on with the resilience of the system at all. So once they go beyond certain limit, that will further increase will become functional redundancy. That is to say, further increase, they become redundant. Doesn't help much except the species itself. Now, if you look at the trophic relationship, in fact, if in the system, the weak trophic relationship is good, just like, for example, if I'm a euphagous animal, I feed on all sorts of animals. Okay, so if A is eliminated, I still got 4 or 5 different species to feed on. And then this is what we call the weak trophic relationship. They actually offer stability, okay, and high resilience. But if it's something like a very tight sort of relationship, the only relationship, just like this old couple, they rely on each other very much. So if you eliminate one of them, the other will die. As opposed to something like one night stands, does not matter. You remove the companion, okay, find another one. Now, coming back to environmental management and conservation. Now, there's several questions that we've got to ask before we manage our ecosystem in a scientific way. The first question is that what kind of resources these animals exploit, and what kind of ecological or biogeochemical process that they regulate, what ecological function and services they contribute, and how important is their contribution. And lastly, equally important is, are they replaceable or non-replaceable? So without them, what will happen? Now, if we can have scientific answer to the above question, and then we can provide a sound argument for conservation of certain species, and then this can also afford us to prioritize species for conservation in a particular habitat, but not just blindly saying, oh, this animal looks cute, and this is why we need to protect them. The other animal looks ugly. Well, does not matter. It's not. Now, so in the recent 10 years or so, there's a major paradigm shift of environmental management. Is from the species to ecosystem perspective. So don't get me wrong. I'm not saying that you should sort of destroy and kill off the species. That's not important. If possible, we should keep all the species. But the most important thing is that we got to know which species is most important, and we got to offer them the highest level of protection. Otherwise, the species that you like will diminish anyway. So in addition to maintaining the rare species or endangered species and overall biodiversity, we should dedicate our effort to ensure that important ecosystem function will be protected. Otherwise, all the target species that you wish to protect may be affected. And then we should also protect the ecosystem function in order for them to provide the desirable ecosystem services and species stay in for us. Now, and this is why recently the major change is that we change from the species perspective to the ecosystem perspective. And the ecosystem management objective should focus on protecting functional group and functional diversity alongside with the protection of species diversity. As I said before, the functional diversity is more important because without that particular species, you cannot perform the function. Now, then the question comes in. It's easy to say theoretically, but in reality, it's very difficult. Let me go through these issues. If you ask how environmental stress and human activity that can affect individual species and community composition, we know a lot about this. You go to have a literature search, there's thousands of papers, thousands of publications, thousands of evidence to show you that in what way, what kind of environmental stress can affect what species and what kind of disaster they can lead to, and they can change communities, composition, so on and so forth. That's not the problem. But if you ask and search the literature, how human activity may affect ecosystem function. Now, there's only very limited study has been carried out, and most, if not all, this study are so carried out in terrestrial ecosystem. Whether these results can be applied to aquatic ecosystem, to marine ecosystem, remain almost unknown. And even for ecosystem function of terrestrial ecosystem is still at the rudimentary stage. Now, so if you want to apply this sort of known scientific evidence to other places, there are only limited study applicable to other places, something like China, tropical and subtropical regions. They haven't done any sort of this study before. So I'm not sure we can use the same sort of principle to borrow these research results on something like temperate region from Europe and from American and apply them to this part of the world. We actually don't know. Now, another important question a lot of people neglect in environmental management is that some species may be very sensitive. Some species may be very tolerant, just like this guy. And then if I ask you to judge, to guess, so this guy, you give him a poison and he can probably survive. This skinny guy, you give him a little bit of poison, you kill him all. That's what we call the resistance. Again, we know quite a lot about this. So that is to say, we know what species are resistant to environmental damage. Roughly, we know about this. But we know very little of whether they will come back or not after the disturbance. Just like this slide. For example, if they're highly resistant, you could disturb them unless the disturbance is very high. Otherwise, they will remain the same. But for some species, you disturb them, and then they will sort of go down, the population go down. But after the disturbance, they will shoot back, come back quickly. Just like bacteria and COVID-19, you remove 99% of them, they'll come back tomorrow. All right? Now, this is again something we do not quite understand. So some species, like opportunistic species, like the barnacles, like the COVID-19, like bacteria, they have a very remarkable ability to grow and recover quickly if we remove a large proportion of them. That's what we call the high resilience. They can shoot back. And then just like this roly-poly, you turn it down, it comes back within seconds. Turn it down, come back within seconds. That's high resilience. But some species, most species with low growth and reproductive potential, they may collapse even if a small proportion of population disturbed. That's what we call the low resilience, like the Chinese white dolphin. A lot of animals, because their reproductive potential is so low. So even if we remove 1/3 of the population, the whole population will collapse because what they call the reproductive encounter. Removing 1/3 of them, they fail to find their counterpart for mating, and the whole population collapse. You don't have to kill all of them. And then the whole population will collapse, and you don't know. It's very difficult to predict. Now, so we have to ask, what are the functional role of the species in the ecosystem? In what way were important to us and our ecosystem? So as I said, Chinese white dolphin, what's the functional role? The functional, what the barnacles, limpets, seahorse, and red tide. So how vulnerable are they to human activity? What is their resistance? And how soon will the population restore itself to the original state after perturbation? The resilience, unless you know the resistance and the resilience and the functional role, otherwise we are not offering any scientific ground for protecting certain species in the environment. Say, for barnacles, you scrap 90% of them, they come back in the next season. Now, so it's not empty works. One good example is fisheries management. We all know that human beings have spent a lot of effort and scientific research into fisheries management. And this is why we work out what they call the sustainable yield. That is to say, we can go and fish, that is to say, at the same time, destroy the environment, destroy certain species, kill certain species. And then until certain level, if you keep on killing certain amount of fish, harvesting certain amount of fish, say 20% of fish of certain size from the environment, it will be sustainable. You keep on doing that, your kid, your grandkid keep on doing that, we still got the same fish, same number of fish in the environment because they can come back. That's called the maximum sustainable yield. In actual fact, for salmon and for pike, we all have data for this maximum sustainable yield. But after overfishing, you kill more than they can afford to, and then the whole system the whole population will collapse and extinct, deplete. Okay, so if we apply this system in or the same method, same approach to environmental protection, that's very good. So that is to say, we can actually have scientific evidence to know how much we can take off, how much we can exploit per unit time. The system is still the same. Oops, sorry. Now, so to sum up, we need the scientific information required to bring us forward if you want to do ecosystem function management. So the question is that what will be the environmental consequences if a given species is affected by human activities? Second question, will ecosystem function ecosystem be compromised? If yes, to what extent? Does it matter? So if we can answer the above question, we will be able to manage our environment based on the science rather than passion, rather than people saying, oh, don't touch anything, don't kill this, don't kill that, and then don't take this, and we'll take everything. We enable the decision makers, stakeholders, and the public to make informed decisions with respect to conservation and sustainable development, not just blindly saying that I like this species, and this is why we need to conserve this. Now, so in actual fact, 5 years ago, China has formed a working group to look at this ecosystem function. And then I was a member, and then we tried to push a major scientific study to answer this question. And then the first pilot point that we use in Bohai, and they start these scientific studies already several years ago. The whole idea is to identify the keystone species and functional important species responsible for energy flow, productivity, carbon sequestration, nutrient biogeochemical cycle in the Bohai system as a model. And then the second task we do is try to elucidate the relationship between biodiversity, ecosystem function, and ecosystem services in Bohai. And then we identify possible human threats to species of high conservation value, keystone species, and important functional species. And then we assess the resistance and resilience of these species to the threat presented to them. So we hope this Bohai case will form a role model for the other part of the world, which never done before so far. And then to show environmental managers how to manage our ecosystem in a rational and scientific way by looking at ecosystem function, not just using the old school of thought, just protecting individual species. Now, understanding our ecosystem in terms of the species composition, function, and services that species provide is important, is fundamental. And then we be able to assess and manage the risk of human activity on biodiversity and ecosystem functioning, and determine the protection priority and development strategy in a scientific way, so that both underprotection or overprotection can be avoided. And for those who are working in environmental management, not only environmental science, we know that both underprotection or overprotection is not desirable. Why? I'll show you an analogy. Say, you're a rich man in your house, you got a bedroom, you got a kitchen, you got a garage, but you need to take care of your bedroom, clean your bedroom, kitchen, and garage every now and then. May I ask you a question? Would you like to keep your kitchen and your garage as clean as your bedroom? Yes, of course you can. You go and clean it up every day like your bedroom, not a problem. Avoid the car coming in, clean your car first, and so on and so forth. And clean your kitchen every day. But is that necessary, right? Because what we call the functional use or beneficial use, the use of bedroom as to go to bed, go to sleep, it got to be absolutely clean. Garage does not matter. You drive your car in and then you walk away. Of course, it can't be too dirty. Same as a kitchen. Likewise, the water quality for protecting coral should be very different from the water quality in a harbor port, because harbor port, your major function is for sort of for hosting the ship and cargo and so on and so forth. So the water quality required is not as strange, should not be as stringent as coral protection of coral reef. Of course, as I said, you can, but whether it's worthwhile or not is you think about this, okay? Because it costs you money, costs you effort. Now, So this ecosystem approach and functional approach will provide a sound scientific basis for us to evaluate the risk and acceptability of development. Whenever you got a development, you know what is the risk and whether it's acceptable, noting that ecosystem function and ecosystem integrity must, at the very least, be protected in all cases. If you destroy ecosystem function, ecosystem integrity, you get nothing. Now, because we have very, very limited knowledge on ecosystem function, ecosystem protection, and so on and so forth, and ecosystem integrity, it is generally assumed, in fact, in Australia, saying that if you protect 80% of the species, would be equal to protect the ecosystem's integrity. But that may not be true. As I said, for argument's sake, if the other 20 species happen just in the unfortunate event, it happened to be the functional important species or the keystone species, you're not protecting ecosystem integrity. So these scientific sort of knowledge would enable us to have a good balance between development and the environment. Now, to summarize, we should manage our environment based on science, in addition to passion. A lot of green group manage the environment or protect the environment based on passion because I like it, okay? The people like different the environment in different way, okay? And it's a very subjective. In addition to conservation of endangered species and species of high conservation value, we must also identify keystone species and the functional important species and accord them the highest level of protection. Otherwise, you'll be protecting nothing. And then we got to work out the species-specific resistance and then the species-specific resilience before we can predict the risk of human disturbance on biodiversity. In the absence of such knowledge, however, the precautionary approach should be used. We should take precautionary approach. We don't know, don't do it for the time being, until we find out all this information. Now, with this, I'd like to come to an end to my talk. And then I hope with this approach, with science as a sort of foundation, environmental protection and the development can go hand in hand together. Thank you.
So thank you, Professor Wu, for this very inspirational sharing on this new thinking about marine conservation by looking at the ecosystem functions. So I would like to invite participants to ask him question or share your own opinion. So you can put down your question in the chat room or just use the raise hand function so that I can identify you, or you just need to speak loud and you can ask question or give comment. So you may start now. Maybe I start with a question. It seems that those keystone species or species with important function may change over time in the same area. Then would this present a challenge in terms of management?
Oh, yes, it's a good question. But I think we are far, far away from answering and from being able to answer your question. As if you look at the literature, look at keystone species, there's only less than, to my recollection, there's only less than 10 keystone species or 20 keystone species have been identified over the world so far, okay? Now, so if you do not understand what is the keystone species in the ecosystem, and then we cannot manage the environment, let alone the question you ask is the second step, and different stage, and then the keystone species may change.
And also I have a question in mind that to understand what we have in the ecosystem will depend on our survey effort. So no matter you're using the trait-based survey or functional-based survey, so given that all the governments in the region with limited resources, how can we overcome this problem?
There's no hard and fast rule in actual fact, and what I hope is that the Bohai pilot study and will publish its result and in another 3 years' time, for example, and then we can borrow a lot of these examples from the Bohai study. The China government actually spent a lot of money, inject lots of money in doing that, and then and that would form an good example. And because of it's also in Asia, and because in Bohai they also cover different habitats, so they may provide a lot of useful information, not necessarily directly applicable, but at least to enable us to have a better guess, I would say. We have a long way, in fact, we have a long way to go. And this is why we call it a paradigm shift.
Okay. So I would like to see any students, participants would like to ask questions or give comments. I saw one on the chat room from Heldai Lakota. She is asking, just like in any impact assessment, we will be asked as to what can be a change in function that is deemed acceptable. Can this be qualified?
Oh, yes.
Question.
Yeah, thank you. And this is again a very good question. So the one who raised the question must be an environmental manager and very pragmatic. And then what if we got to find, if we predict some change in the ecosystem function, and then that's deemed to be acceptable or not acceptable, it depends on to what extent and the beneficial use of the site in question. For example, if I may cite an extreme example, if you want to protect the coral reef, any change in the ecosystem function of coral reef, well, may not be acceptable. But the same change may be acceptable if you do on a swimming beach, and then the larger change can be accepted or become acceptable in sort of a harbor, for example. But again, is a question which is not only related to science, but also a lot of the human judgment and depends on the culture and so on and so forth. It's extremely complicated.
So it also depends on this beneficial use you just mentioned.
Yes. So yes, you have to make reference to the beneficial use that you decided.
Yeah. So any other questions? Or Hyundai, actually you can speed up. So you can show your face and talk to Professor Wu if you like.
Good afternoon, sir, Professor.
Yeah.
Is this okay?
Yeah. Hello.
You can hear me?
Yes.
Because this is something that we are always, we often see ourselves under debate because there are different perspectives in economics, in ecology, and well, those are the most important fields that usually would go into debate for this. And if it's economics that comes into play, the ecological explanations usually are shelved and they're left behind. So your perspective actually on lobbying for we must happen already. Say there is so much development in the new normal. I think it doesn't happen only just here in the Philippines, but elsewhere as well. So for it to be effective, it has to be passed as a law.
Yeah.
And before it becomes that, there'll have to be many cut-offs. There'll have to be a lot of balancing that has to happen.
Yeah. Yeah, I totally agree with you. And then, in fact, as I always say, the environmental management is a very complex issue. And then people, I told my students, and then doing environmental science, if you think environmental science is almighty, you're wrong. And then, normally, under most, if not all circumstances, environmental economics is more important. Well, as I said, I mean, you can actually keep the environment as clean as your bedroom anywhere. It's how much effort, how much money you'd like to put in. And we can, in fact, remove all the waste from our wastewater, right? And then up or down to the level, just apparently as this kind of water. It's a matter of money. And then economics will be again override by environmental politics. Well, you get the politics, a lot of these are not actually considering environmental economics or the environment, but rather politically, they think it's correct. Okay, and then just like a lot of these minority, something like in the US, for example, they protect the Indians, okay, at any cost. But the same form of protection will not afford to the other. Likewise, in Australia, and the government is prepared to put lots of money and to protect, to cater the need, the environmental need for the Aborigines, but not for the others, mainly political reasons. So again, it's a very complicated issue, and yes, and this is why it's a highly interdisciplinary and topic.
Thank you very much, sir.
Yeah,
Thank you very much for the discussion. Okay, once again, thank you, Professor Wu, for giving two very insightful talks today. And he will give another talk tomorrow. We're looking forward to. So now, may I invite the next speaker, Leifen Jin Nin. He is come from the Polytechnic University of Hong Kong. So let me see my script first. He is currently an assistant professor at the Department of Civil and Environmental Engineering at the Hong Kong Polytechnic University. His main research area includes air pollution and human health, airborne and foodborne transmission of antimicrobial resistance and pathogens, and pollution-induced immunosuppression in Chinese white dolphins. He using the Chinese white dolphin cell lines he isolate and culture in the lab. Today, he will provide training under the topic of toxicity assessment of chemical mixture. He started this area of research when he was doing his PhD in Queensland University under Beata lab. So now may I invite Nathan, please.
Thanks, Kenny, for your kind introduction. Today, I will briefly talk about some concepts and some application, okay, very briefly, and try to make this complex mixtures simple, okay, for as a training purposes, okay. So actually we're experiencing multiple chemicals in our daily life. So a very good example is actually what is good for tonight, okay, Friday happy hours, the cocktail, okay. So which includes a lot of ingredients, right? And the combined effects of these things makes our joy, brings joy for us, and also if too much, okay, if too much, there may be some cause some health problems. All right, so this is the same, which is also true to the environment. Okay, similarly, our marine environment, aquatic environment, drinking water sources, all sorts of things, our wildlife, our human bodies are exposed to on the bad side. Okay, it's experiencing this kind of multiple stress of the and synthetic and natural organic chemicals, as well as their transformation products, which may be formed after being released into the environment. Okay, some including what we have known as personal care products, right? Pesticides, herbicides, yeah, natural hormone, and some mimics, right? And well, just to name a few. Okay. Now, so as we know, so in the commerce, there are more than 100,000 chemicals are there already. So how much have we really known on the front? Think about an iceberg. Okay, this is the chemical universe. So how many have we really known? How many have we known? Okay, so but the important thing is, do we really need to identify them? Okay, do we really need to identify those unknown? Based on what? Now, this is the main questions I want to bring forward at the very end, in the very beginning. Okay, so now, so there are two views. Okay, two views of chemical mixtures separately.
So, Dr. Jin, are you sharing your PPT?
Yes.
But we cannot see it.
Oh, sorry. Is it right now? It's okay now.
Yes.
Okay. Then maybe cocktail. Okay. So the chemical universe in the world. All right. All right. So there are two views. Okay, from a chemist's view and the toxicologist's view about what mixture, the perception. Okay, on mixtures. So chemical analysis, chemists. Okay, develops a different instrumental analysis to measure metals or whatever on a target way, conventionally, to analyze those regulated chemicals, okay, or what they want to measure to their interest. So it's quantitative for key target chemicals, right? But there are also unknowns with the difficult and work intensive to identify, but it can be, okay, solved, okay, with the advances of so-called non-target analysis. But from a toxicologist perspective, okay, we are really trying to fully integrate these chemical groups, okay, to summation of groups of chemicals with common mode of action. So the basis is on what we really view are, so on the toxicology perspective, like dioxin-like chemicals, which are acting commonly on area hydrocarbon receptor, or those chemicals all together working commonly acting on oxidative stress or as hormone mimics. But so the toxicology try to have an integrated mixture effect measurement, okay, mixture effect measurement using a common endpoint. But this is an integrative way, but the drawbacks is the single chemicals cannot be resolved. But how can we really complementary? But actually, these two can be complementary to each other to solve the question, to solve two major questions. So what are the combined effects of all sorts of chemicals in the environment, in the water, in the sediment, right, in our food, or also, and the other thing is very important is now how much effects can be really explained by identified chemicals, or for example, what we analyze can explain how much of the total effects, so that we can prioritize individual chemicals, whether they are important or of lower importance. Okay, so then now, as we said, we based on this kind of toxicology perspective, we need something to measure on the endpoint. Okay, so the adverse outcome pathway and the toxicity pathways provide this kind of conceptual framework, so that we can now. So with advances of 21st century toxicology, now we know this kind of cell response up to higher level of our organization, up to ecosystem health. This is a promulgation of biology effects. Now, as Professor Wu Professor Rudolph Wu just talked about, so the ultimate goal, okay, the ultimate goal, protection goal, is targeting the ecosystem health. Okay, so but they're very difficult to measure. Okay, but they are very difficult to measure. Now we can do something, okay, with low complexity acids for invertebrates, like copepod that commonly used, that Professor Kenneth Learn are experiencing. Okay, so Now, but we can also down to this level, cell response, which can be an integrated bioanalytical tools can use as an early indicators of hazard potential of chemical mixtures and are indicative modes of toxic action, like targeting this kind of initiating events. Okay, as we said, for example, induction of xenobiotic metabolism, or some more specific receptor mediated effects like estrogen, right? Those hormone receptors, right? And also some reactive toxicity like DNA damage, inflammation, oxidative stress. Okay, then all the way up to some apical events like cell death. So we can do this kind of cellular toxicity, cellular assay, as well as some low complexity like algae toxicity assay. Okay, so can be used in a high throughput way to allow us for more rapid screening of the mixture effects contained in complex environmental samples. So this is a typical workflow. Okay, typical workflow. Like we start from dispensing of those target cells we want to measure, right? Prepare the dosing place, and we treat the cells with the individual chemicals and mixtures of chemicals, as well as environmental complex mixtures containing unknown chemicals. Okay, then we do this kind of so-called dose response analysis, concentration effect analysis, and measure the endpoint, like using some reporter gene or enzyme analysis to measure those kind of responses as shown here. Okay, now before we move on to more complex mixture, okay, toxicity prediction or whatever. So here we need to first understand, okay, very important concept of this kind of concentration response. Now this is actually from a pharmacological perspective, in the Paracelsus, or the very Asian Italian, okay, Italian pharmacist, okay. So his famous notion is all substances are poisons. So there's none which, so only the dose that makes a poison or a remedy. Okay. So basically, we do this kind of concentration response or dose response. The x-axis as the concentration, okay, increasing concentration of a substance, okay, for example, pesticides, right? And then the effects, the y-axis is the increasing effect of, for example, the cell death, inhibition of photosynthesis, right? In the case of photosynthetic pesticide inhibitor, okay? So we do not for mixture toxicity evaluation, or very important that we need to have certain benchmark, okay? Like so-called EC50, effective concentration of 50, which means the concentration at which 50% of the effect, okay, of the maximum is occurs. Also, we may use EC10, okay? Now, there are also two very important metrics, so-called no observed effect concentration or lowest observed effect concentration within this kind of the control range. Now, we will use this as an example for later. They're very important concepts just to bear in mind. Okay, later we will talk about something from nothing. Even those chemicals at their no observed or lowest observed effect concentration mixed together, they may still produce something, okay, so-called something from nothing, right? Now for this kind of concentration response evaluation, this software, okay, commercially available software called GraphPad Prism is just a really good tool to allow you to fit this kind of nonlinear sigmoidal equation. Okay, so which it is also visually appealing, and it has great handbooks, right? But you must bear in mind that for modeling, okay, trash in, trash out, garbage in, garbage out, it it does not free you from understanding what you are doing. Okay, we need to understand the basic concept. Right. Okay, now we have talked about how we evaluate individual, okay, individual and the chemical. Then we think about the mixture. Okay, there are the basic concepts of mixture toxicity. So how a bunch of chemicals act together, okay? How can we predict them? And there are some basic concepts behind. So the joint action of chemicals depends on what? Depends on the site of action, target site, and the mode of action, which we have explained here. Okay, specific estrogen mediated receptor, right, or whatever. Okay, so now basically there are 4 categorizations. If the chemicals do not interact with each other, and they act on the similar joint on the same target site, for let's say target on this a group of chemicals, all commonly targeting the area hydrocarbon receptor or targeting the hormone receptor, the same hormone receptor. So eventually they will exhibit a similar action, okay, joint action, so-called concentration addition, okay, in the follows this way. Now, if those chemicals are working on different target sites, now one is working specifically on photosynthesis inhibition, one is non-specifically targeting cell membrane, but when they work together on algae, okay, for example, on a plant, so they may display, okay, dissimilar joint action, which can be described by the so-called independent action ion model. Now, these two modes can be mathematically described, okay, but more complex things happen if some chemicals, they may have some interactions are present. They may have more, but still on the same site, but they will exhibit a complex similar action. Okay, but for those interaction that are working on different target sites, they may have dependent action. But from the literature, okay, so many decades of research. So these two are quite rare, okay, in the environment. So most common modes are these two, and concentration addition, okay, we can guarantee that it explains very well, okay, explains very well the joint impacts of a bunch of chemicals in the environment on certain biological endpoints happen in that environment. Okay, now this is a typical example. Okay, back to it's not 2 decades ago, right? Working on these authors working on endocrine disruption. Okay, trying to understand the eight estrogen receptor activator. Okay, so all individually they are below their threshold level, but once they mix together as experimentally determined here, so it is very close to what can be predicted from concentration addition. Okay, now because simply they are working on the same target. Okay, for example, in this case, it's estrogen receptor. It's a hormone mimic, right? So there are also a lot of other examples, okay, for something from nothing. All right, so for chemicals with the same mode of action, I think we don't have to go through the details because we will share this slides later. Okay, also something from nothing for chemicals with different modes of action. Okay, described by independent action ion model. Okay, so just a little bit a wrap up. So independent action, okay, is strictly valid, okay, only for compounds with different modes of toxic action, and in most cases different targets, okay, target size. And theoretically, in principle, if compounds with dissimilar modes of action are mixed, each in concentration below threshold of effect, the mixture cannot have a significant effect. But in practice, it's not that. So And also for CA model, okay, typically holds valid for chemicals with the same mode of action. So if compounds with the same mode of MOA are mixed, each in concentration below threshold of effect, so the mixture may have a significant effect, okay, which you have seen from the 8 compounds for the estrogen estrogen receptor induction, okay, and also which is also independent of the mode of action. Most apply the concept in ecotoxicology because as we have seen over the decades, so the mixture effects in the environment, okay, triggered by known chemicals really follows this model, okay, which makes life easier, okay, because it's mathematically describable, okay, in a simple equation. So for these two, okay, so quite rare. So I don't have to repeat it again, okay. So think about now. Then we really need some quantitative thinking because talking about prediction, okay. And so think about 2 component mixture, chemical A and chemical B, okay, when they mix together, okay, so-called binary mixture. So here is a top concept is so-called Tox unit. Now, so we use this kind of isobode diagram to describe, okay, what is under what kind of situation. So these 2 chemicals can act on a concentration addition way, which also when it goes for antagonism or independent action or synergism, okay. So more than additive. Antagonism means less than additive. Okay. So the toxic unit TU. Now think about it. We do those concentration response for each of the chemical A and chemical B, and with a derived effective concentration, let's say EC50. Okay. So then we, for example, take 3%. No, sorry, 30%. Take the chemical A at the concentration of 30% of the EC50. Okay, then take 70%. Take another chemical, the chemical B at its concentration of 70%. Okay, the fraction needs to be the summation of these, because 2 chemicals, one 30% or 70% or 50% or 50%. Let's say the summation as 1. Now, if the toxic TU1, okay, toxic unit plus toxic unit, the summation of the toxic units of these 2 chemicals equals 1, then we can say they strictly follow the concentration addition, okay. But there are some cases these two plus sum together may be greater than 1, okay, so which is less than additive, and or greater than 1 less than additive, or less than 1 is so-called synergism, okay, more than additive. Now there are some example of synergistic interaction of, so for example, a very traditional example of atrazine, a banned herbicide, can in interaction with organophosphate pesticide, okay, so they may exhibit the synergistic more than additive, okay. So the hypothesis behind is maybe the atrazine induces enzyme that catalyze the activation step and thus produce more active agents. So the combined effects of these 2, okay, pesticide and pesticides and herbicides act more than additive, okay, but they are very rare examples, especially in the environment when it is diluted in so many chemicals altogether, okay. So the concentrations addition still hold, okay. Now then think about it. If the components are expanded, okay, from 2 to 18, let's say, okay, in this case, the 18 atrazine analogs, okay, are all inhibitors of photosynthesis by binding to photosystem 2. So with a common target, common mechanism. So we do this kind of prediction, okay, we predict the joint effects using the independent model, okay, independent action IA model, we also do the CA prediction, which is the dashed line. Okay, then we really mix together and test it on the algae. Okay, and see the photosynthesis combined effects of the 18 chemicals. So it really follows very well for the concentration addition because they have the common target, right? So how about independent action? Okay, so now 16, in this case, 16 dissimilarly acting substances. Okay, some specifically targeting the photosynthetic system too. Some are non-specifically targeting other parts of the algae system. Okay, so they pretty well follow the independent action model. All right, so now this is not only the mixture. How about the mixture? Typical environmental pollutants in the real environment. Okay, so they are not only applicable for mechanics endpoints and the low complexity system as we just talked about before, okay, and for the algae, but even for long-term sublethal effects in fish in vivo. In this case, is an induction of betalogenin, okay, in male fish as a measure of estrogenic activity. Okay, so with natural and artificial estrogens, okay, like estradiol E2, which is naturally occurring in fish and in humans, right? And also a contraceptive, like a synthetic hormone, the estrogen mimic, so-called E2, okay, mixed with xenoestrogens, right? These, now we have known this kind of bisphenol A, okay, they act in mixture additively. This is a very typical example, okay. Then also, now, these chemicals are artificial designed mixtures, okay. Then we talked about all sorts of environmental real mixtures of chemicals occurring in the environment. Now, how about we mix the chemicals, okay, those regulated chemicals, okay, in the under the drinking water guideline values in their ratios of the drinking water guidelines, as well as mix those chemicals in their ratios of their occurrence, their concentration ratio in the real drinking water. Now, so we test them on the simple bioassay, so-called microtox assay. It's a bacterioluminescent assay, okay, to measure the chemicals disturbance of the membrane, okay, and kind of as a measure. Okay, so we do this kind of equipotent mixtures and mixtures in ratios, okay, in the drinking water guidelines or as the occurrence in the real water sample. So basically all are very close to CA. Now also another example is for surface water, right? So the mixture model, toxicity model of concentration addition has very good predictive power, okay? But specific chemicals drivers of mixture effects, right? And also different chemicals for different endpoints, okay? So we will talk about how we can really identify or quantify the contribution of known identified chemicals to the mixture effects in water or in biological samples. Okay, we will talk about those algorithms later. All right, so now, as we know, okay, the aquatic environment contains a diverse range of contaminants and transformation products. Okay, so if we inject a water sample or inject a sediment sample, biological tissue sample, okay, into the GCMS or air in our instrument, okay, typically we will identify 20,000 peaks, okay, many of them unknown, with unknown identity and quantity. So so in vitro bioassays can complement the chemical analysis to measure the entirety of the cocktail effects, okay? So the mixture effects by in vitro bioassays quantified, but quantitative agents not identified. So that's why we need to better understand the link between chemical analysis and what we can identify, target or non-target, and what we can measure for their combined effects. So, and also need to think about the mixture models that we can use. Is it applicable for, so for example, the concentration additive? In the next part, very short introduction about the so-called iceberg models. So to help us quantify, quantify how much of the identified chemicals, known chemicals, contribute to the whole mixture effects. Now, so in a case analysis from the Germany, so those scientists sampled water samples, surface water samples, along a gradient, along with a gradient from upstream, relatively pristine, and to the outlet of the wastewater treatment plant to collect the effluent, and all the way to the downstream potential with dilution, but still maybe to some extent being affected by this wastewater treatment plant. We do this kind of effect analysis, bioanalysis, for photosynthesis, algae toxicity, or oxidative stress, or whatever, to characterize the mixture effects represented by the whole iceberg. So we derive this kind of EC for sample, so-called tox unit of the sample. And also we detect individual chemical concentration, especially in this case, it's target analysis for regulated chemicals under current regulation surveillance. Then we also tested these chemicals in the bioassays to derive the effect concentration. So we can summarize summation them to for the toxic unit of the chemistry, toxic unit of the identified chemicals as the tip of the iceberg. So we want to know how much of the tip have we really seen, which means how much of the identified chemicals, how much of the entire effects can be explained by the identified chemicals. So for chemical analysis, they follow the list of the regulated chemical inventory. So they use this kind of ARC mass analysis to quantify the concentration. So pretty straightforward. And then they also fingerprints of the single chemical compounds for their effect potency, to characterize this part, so that we can characterize the mixture effects of known chemicals, So in this case, like for example, photosynthesis inhibition, right? So the majority of the identified herbicides can explain the majority of the mixture effects. So we can also prioritize according to their contribution, prioritize diuron or terbutalyl azine explain most effects. So which means they are the greatest risk drivers. Number 4, the mixture effects on the fish embryo or oxidative stress. So the identified chemicals only identified regulated chemicals only explain a very small fraction of the entire mixture effects of the water samples. All right, this is another example during my PhD. We do this similar analysis for turtle. We extract use a so-called PDMS polymer to extract the mixture of chemicals, right, from the tissue, like fatty tissue, fat, and then also the turtle. For turtle, it's blood. So we treat those extracts the entirety of the chemicals, then those in the cell-based assays for every hydrocarbon, like for dioxin-like effects, and also as well as for those adaptive stress responses, like oxidative stress, DNA damage information. So we also quantified a so-called persistent organic pollutants in a legacy way. So like the dioxins, pHs, PBDEs, right, PCBs. Now all those chemicals, can explain very well for this specific, the receptor-mediated effects, but it's still explain very small fraction of the oxidative stress. So we have seen two types of iceberg mixtures. Okay, so for those biologic effects that is receptor mediated, okay, so which can be targeted by a very small number of highly active chemicals, so they can explain almost the entirety of the effect. So then this one is much easier to regulate. But for those non-specific effects like cell death, right, or oxidative stress, so the unknown, typically we see the tip a very little fraction, so which means many chemicals contribute to the effect. It also triggers us to think about how to identify the unknown mixture toxicity contributors with advances of so-called effect direct analysis. But in this lecture, we won't detail it due to the time limit. As well as thinking about, now, if we cannot really identify everything, then can we really regulate the mixture effects, okay, beyond the regulating the single chemicals? Now, this is also another advance, so-called effect-based trigger values, especially for water side. Okay, they're advancing, but for sediment, for biological, wildlife health and human health, it's not advancing that far. Okay, so some take-home messages. Okay, so the cocktail, although we experience very complex chemical universe, okay, in the environment, but the life it can be made easier, okay, by this kind of simplicity. The cocktail effects of chemicals in the environment, okay, follows generally, okay, the concentration additive matter, and also mixture toxicity modeling, okay, combining chemical analysis and bioanalysis can help us to quantitatively prioritize the mixture risk drivers in complex environmental samples, okay, in marine, in a terrestrial system, okay, and also in fact, also there are two advances we need to think about. When do we need to identify the unknown effect drivers? Also, do we really need to regulate mixture effects beyond the single chemicals? Okay, thank you very much for your listening, and welcome your questions and comments. And here is my email, okay, for future response.
So thank you, Dr. Jenny. This is a very difficult topic. I would imagine some of the participants may have questions. Any question from the participants? You're welcome to drop your question in the chat box or just speak loudly. Any questions? This is still ongoing research on mixture. It's a very hot topic.
Very hot topic and very challenging topic. So also Professor Kenneth Leung is also leading a very large project on this issue, right, in the marine system. So we're looking for future, now for your funding success, okay, as well as for our future collaboration. As we also welcome the collaboration from the Southeast Asia, okay. Think about those marine water quality, okay, in terms of mixture effects, as well as for marine wildlife.
So everyone seems very quiet.
Sorry,
yes,
sorry,
please.
I have a question.
Yes, please.
Oh, hi, Dr. Jing. Thank
Yeah,
you
please.
very much for your presentation. And I have a question about the mixture toxicity. And you talk about the chemicals don't have interactive effects. But what about the chemicals that have interactive effects? What kind of mixture toxicity model, or how can you just study the mixture toxicity of those chemicals that have interactive effects?
Oh, thank you very much, Xia Ying. Okay, so now first I should say from the decades of research, okay, there's one literature review summarizing, compiling all sorts of mixture effects in terms of different joint actions, okay, from synergistic effects, antagonism, or concentration addition, or independent competition. So far, we have seen synergism, okay, has very rare cases, okay, there, because this is actually from the an experience from the pharmacological side, okay, because they normally use only at very high concentrations, these kind of interactions or more than additive, okay, effects can happen. For example, typically for pesticides, like which I have shown this example, that like atrazine plus the organophosphate, because maybe the atrazine activates one enzyme that can catalyze the organophosphate into a more potent, more toxic transformation product. In that case, they may act together more than additive. But these are very rare examples, okay? And also think about in the environment, those chemicals are normally present at very low concentrations. And also think about the amount of chemicals, 1,000, more than 10,000. So everything, because of this kind of dilution effect, even those things can act very strong interactively, may be diluted, okay, very likely to be diluted in the presence of thousands of chemicals, so that they eventually may still act on a concentration additive way. Okay. And we may have further discussion later.
I think
Okay.
I would like to follow up. It means you support Michael Wong's proposal on this funnel effect.
Yes.
When the mixture increase to certain numbers, say 50 to 80, then they will behave just addictive, right?
Yes, I have some. Now, I haven't shown some preliminary result here. Okay, so actually, I have tested up to 200, no, not 2000. Okay, 200. So starting from two binary combination, 2 components, up to 200 components. So the more the chemical the greater number of the chemical combined together, so the better the concentration additive model can predict for their joint effects.
Oh, have you published this work?
No, not yet.
Very impactful research.
Not yet. So I want to publish it with you, okay, in support of your research.
Very good, very good.
Yeah. So, but it's very hard. Okay, we also need to have those kind of automated and robotic platform if we really want to go to thousands of chemicals.
I see. Yeah. Very interesting.
All right.
Saying, sorry, you have follow-up question, right? Saying, Dr. Xian. You need to unmute.
Sorry. Oh, sorry. I mean, what about the microplastic and the other organic chemicals? I think
this is another issue. Okay, this is another issue. So here we're really talking about pure chemicals.
Okay.
So what you are really mean is you mean is really about multiple stressors. Okay. Because this kind of particle effects plus the chemical effects, how can we predict that? It's another level of complexity.
Okay.
Now also think about more complex physical chemical parameters, if temperature, right, humidity, all sorts of things on climate change or whatever, okay, then it will be even complex. So we try to address these chemical, okay, mixture effects first, and then we go up to a higher level of complexity. But we really enjoy it and also hope to collaborate with each other, okay, in the future.
Thank you.
To develop a better predictive model.
Thank you once
Thank you
again,
very much.
Dr. Jenny. So very, very interesting study. So we will take maybe perhaps a 5 minutes break. We will return by 3:45, 3:45. Then we will have Dr. Phoebe Warren to talk about chemical pollutants of emerging concern. So we will take 5 minutes break. Okay, I will see you return soon. Okay, we take a break. Bye-bye.